challenge was to align the different developments in the different work packages, for example. So we have really progressed in the individual work packages and then already trying to bring those results together and actually combine, for example, the production of the raw materials with the first testing results, processing of the material and so on.
Our biggest achievement, I think, has been cultivation of scurvy grass last year, and we were able to upscale it up to 1,000 liters, which was a gradual process that included multiple smaller scale bioreactor cultivations.
Identification of new plant species that are being now processed through our scientific track, scientific research, and so now we are digging more into interesting molecules and biological effects of these new plant species.
Bioreactors has several advantages for the environment. We are not, let's say, impacting the environment, like growing them in the open space, subtracting fields to other purposes such as agriculture. And also we can control the growth environment of these plant cells. The technology is absolutely scalable, it's continuous, so it allows to mass produce plant cells within a relatively short amount of time if the bioprocesses are well optimized, and then to process the obtained biomass to get phytochemicals or other type of added value compounds for different industrial applications. And also it is contaminant free and continuous, so this responds quite well to the requirement of specific industrial applications such as cosmetics, but also other type of applications such as pharma or medicinal applications.
So one of the special things associated with the plant culture system we are using, and this is aeroponic cultures, is that we can alternate within a period of time. We can alternate a period where the plant is kept in ideal condition for growth, and then after this period we change the cultivation conditions and we stress more the plants, and at this time the plants will react in producing more compounds of interest. So basically we have one, shaping the biomass and producing plants in large quantities, and two, producing interesting molecules once the biomass has been shaped.
At VTT we are working mainly on chili cultivars as well as in the rowan and scurvy grass. Also we got some samples from our partners, and it includes the kochia and menta, which we currently developing cell lines for them.
It's not very simple or easy. You cannot just do the same but in larger scale, but you really have to optimize every step on the way and also have a clear understanding of the physics, for example, how the shear forces work in this larger bioreactor environment.
The idea during the scaling up is to first to maintain the biomass production. This is really important because usually we observe some decrease in the biomass yields. So the idea is to maintain all the parameters that we are using at laboratory scale, and to maintain all these yields across the upscaling phase.
Next achievements to come for us are probably related to the valorization of co-products, or the so-called upcycling strategy, where basically we need to use all the plant parts, not only the plant part that we are focusing on at the origin, in our case it's roots, but of course aerial parts of plants like stems and leaves have also a lot of value in terms of molecules and activity.
Okay, so we are now halfway in the project. 2 years are ahead of us still, and I think there is quite some deliverables and some milestones that we can achieve in the forthcoming 2 years. Pre-commercial evaluation will take place, so the feasibility, the time, the cost can be evaluated, so we can produce data that can be of interest. We are comparing different production systems, and of course, within the partner participating in the work package, we are exchanging material. So it is possible to compare one species across different production systems in cell cultures, heavy roots cultures, or plants growing in aeroponics or in the field, and we can actually have a unit of measure across the different production systems for the same set of molecules or different molecules, and then choose the most appropriate production systems to meet cosmetic application requirements.
To be honest, I think a lot. Currently, we are at VTT, we are working with several plant cell cultures as well as organ cultures, so-called heavy roots. We have already analyzed, chemically analyzed the cell cultures and did antimicrobial tests and bioactivity assays, either at VTT or with help of our partner. And we find very interesting results. For example, we found out that our chili cell lines produce antimicrobial compounds because we saw the inhibition of the bacteria. We are planning to upscale these samples, the cell cultures, and monitor again the inhibitory effect of the extracts, which means in a way that we could produce more of the target compounds. We are looking forward for more successful bioactivities.
Keeping in touch with the very best of European research, especially academic institutions, is very important because it helps us staying on the wave of innovation and keeping as much as possible innovation at a very high level.